Hello and welcome. In our lesson today, we are going to discuss high-order thinking questions regarding cell physiology. So these are simply the application questions. Now, I know that some students find application questions a bit challenging, but with the current trend in setting of biology papers, these are just here to stay. So if you can manage to get as much practice as possible, then you stand a better chance in improving your performance in biology. So in this lesson today, we are going to tackle application questions on the three physiological processes: diffusion, osmosis, and active transport. Now, I want to say this. If you're interested in attempting the questions yourself at any point, kindly pause the video, engage with the questions, and then proceed with the discussion to determine whether you answered them correctly. All the best. Let us start. Our first question: for each of the following processes, state the physiological process that is involved. Just a reminder, we only have three physiological processes, so it's up to us to determine whether osmosis, active transport, or diffusion is involved. So part A, absorption of water by plant roots from the soil. Now, whenever you have water molecules involved, movement of water molecules involved, then that cannot be any other process apart from osmosis. So automatically A becomes osmosis. Part B, absorption of mineral salts by plant roots from the soil. Now, mineral salts from the soil can be absorbed by the roots of plants either by diffusion or by active transport. Now, either of these two answers is correct. Now, the reason is it depends on the concentration of the mineral salts in the surrounding soil. If the concentration of mineral salts in the soil is higher than that within the cell sap of the root hair cells, then the process absorption occurs by diffusion. But if within the cell sap of the root hair cells, the concentration is higher than in the surrounding cell, what happens is that the mineral salts will still be absorbed, but against the concentration gradient. And this, of course, is by active transport. Part C, absorption of glucose into the bloodstream of animals even when the blood has a high concentration of glucose. Now, what happens is that after digestion is complete at the ileum, the end products of digestion, simply meaning glucose, fatty acids, and glycerol, so on, need to be absorbed into the bloodstream. Why? So that they can then be transported to the rest of the body. Now, when absorption of the end products of digestion takes place, for example, glucose, what happens is that it reaches a point whereby the concentration of glucose in the bloodstream is higher than in the villi. So, glucose will still be absorbed, but in this case, again, it's going to be absorbed against the concentration gradient. So, active transport is involved. Let us now proceed to the next question. The diagram below is an illustration of a certain physiological process that occurs in cells. Now, let's look at that diagram. Now, if you look at that diagram, by the way, you can clearly tell that this is a cell membrane. How so? Because we have the double layer of phospholipid, we also have the protein molecules. Now, these are the blue structures. So as you can tell, in between the two protein molecules, we have an opening that allows the particles to pass through in the inner part of the cell. Now, part A, name the physiological process illustrated by the diagram above. Now, we are given hints from the diagram on which process this. Let's look at the concentration of the particles. We are having particles moving from a region of low concentration to a region of high concentration. So that clearly takes out diffusion and osmosis. It cannot be these two processes. Why? Because in the case of diffusion as well as osmosis, we are having molecules or particles moving from a region of high concentration to a region of low concentration. Whatever is happening in this diagram is the reverse. Particles are moving from a region of low concentration to a region of high concentration. And that, ladies and gentlemen, can only be active transport. Now, there's actually another hint, and that is the requirement of ATP. So ATP here stands for energy. Okay, so diffusion and osmosis are passive processes. That means that they don't require energy naturally. And the reason for this is because particles move along the concentration gradient. Active transport, on the other hand, causes movement of molecules from regions of low concentration to regions of high concentration. Now, this moves against the concentration gradient and therefore requires energy. And those are the two reasons for why we are stating that this has to be active transport and no other process. Part C. What will be the effect of treating the above part of the cell membrane to temperatures above 50 degrees Celsius? Give a reason for your answer. Now, the cell membrane consists of protein molecules which are sensitive to changes in temperature. That means that if you were to either decrease the temperature or increase the temperature, it's going to affect the functioning of the cell membrane. Now, in this case, we are being told that temperatures are going to be increased above 50 degrees Celsius. This, of course, will end up damaging the structure of the cell membrane. And by doing so, it will prevent the cell membrane from functioning. Our next question, a group of students took 3 whisking tubings labeled X, Y, and Z and placed 3 different sugar solutions in each. So there we have our whisking tubings with different sugar solutions in each. This simply means that each whisking tubing has a different concentration of sugar solution in it. The whisking tubings were then placed in a beaker containing 10% sucrose solution. The setups were left for 5 hours and the results are shown above. So on the diagrams above, we can clearly see what was present at the beginning of the experiment and what happened during the course of the experiment. Now, just to remind ourselves, within each of the 3 whisking tubings, we are going to have a sugar solution. A sugar solution simply means that sugar particles were dissolved in water. Now, the concentration of this sugar solution that was placed in X, Y, and Z was different. Now, these 3 whisking tubings were then placed in a beaker containing sucrose solution. Sucrose solution is simply sugar solution. Now, this can seem a bit confusing, but essentially what we'll be doing is that we are going to compare the concentration of the contents of the whisking tubing to that of the beaker. So we'll be comparing 2 sugar solutions in each case. The sugar solution present in whisking tubing X and the sucrose solution present in the beaker. And then we'll move forward to X, do the same. We'll compare the concentration of the sugar solution in Y with that of the beaker. And why are we doing this? Because we can clearly see that there's a process that is taking place, because for 2 of the whisking tubings, there's an increase in one and a decrease in the other. So we are going to have movement of water molecules in both of these cases. So we need to determine the concentration of these 2 before we can proceed. Part A, identify the process being investigated in this experiment. So in this experiment, we are going to have movement of water molecules, and that only ever occurs through osmosis. Part B, account for the observations. So the question is simply asking us to explain the observation, meaning that our observation is with regards to X, we are going to have a decrease in the solution in X. So we'll have to explain the reason for this. In Y, no change whatsoever. So what happened here, or what didn't happen? And then the last one, Z. Z has an increase in solution. So we need to account for this too. Now, when discussing osmosis, I like to break it down into 3 main steps that students need to incorporate in their explanation. Part 1, determine the concentrations of the 2 solutions. That simply means that determine which solution is hypertonic and which is hypotonic. Now, this step is very important because water molecules will always move from a region that is hypotonic to a region that is hypertonic. Now, in case you find this a little bit confusing, please refer to my video where I discuss this in detail. Now, once you've determined the 2 concentrations of the solution you have, the next step, of course, will be the movement of water molecules. And this is easy once step 1 has been done. The last step, what are the effects of these movements of water? So you tie these 3 together and you end up having a perfect answer. Now, let's start with X. Now, in the case of X, there's a decrease in the solution in the whisking tubing. The concentration in the whisking tubing is hypotonic. Now, remember, water molecules will always move from a region that is hypotonic to hypertonic. So that means that if we are talking about a decrease in the water molecules moved from X into the surrounding solution. So that's how we're going to start out our answer. The solution in the whisking tubing X was hypotonic to the sucrose solution. Now students, never say that the whisking tubing X was hypotonic. This is wrong. Whisking tubing is just a membrane. It does not have any concentration whatsoever. So it's actually the solution in the whisking tubing that is hypotonic to the sucrose solution. Next step, movement of water molecules. Water molecules will move across the semi-permeable whisking tubing and into the sucrose solution by osmosis. Always mention that the net movement of water molecules is a result of osmosis. Now the last part, what is the effect of this? It leads to a decrease in the level of solution in the whisking tubing. And there we have it. Now let's proceed to the second whisking tubing. Why? Now we can clearly see that there has been no change to the contents of the whisking tubing. It is the way it was at the beginning. So that means that this solution is isotonic to the sucrose solution. Now when we compare the concentration of two solutions and we find them to be isotonic, that simply means that there is equal concentration, or the two solutions have the same concentration. Now, for osmosis to occur, there needs to be a concentration gradient. So if there's no difference, then that means that osmosis does not take place. So how do we phrase this? The solution in whisking tubing Y was isotonic to the sucrose solution. Therefore, there was no net movement of water in either direction. So what are the effects for this? No change in the level of solution in the whisking tubing. Last one, Z. Now, with Z, there's actually an increase in the solution in the whisking tubing. Now, let's remember our point. Water molecules move from hypotonic to hypertonic. So, if Z is gaining water molecules, that means that it's hypertonic to the sucrose solution. So, the solution in whisking tubing Z was hypertonic to the sucrose solution. Therefore, water molecules moved from the beaker and into the semi-permeable whisking tubing by osmosis. What are the effects to this? An increase in the volume of the solution in the whisking tubing. Now this is a very good application question on osmosis. I hope that it was well understood. And that, my dear viewers, brings us to the end of our lesson today. Be sure to check out my other video where I delve into more complex questions regarding cell physiology. See you there.